Good morning. I'm Dr. Sherry Fleming, the director of the Johnson Cancer Research Center. I would like to welcome you to Open House, and we'd like to show you some of the labs and undergrads and what they're doing for cancer research. We have a number of members, over 90 members from across campus, from health and human sciences to engineering to arts and sciences, ag, and even vet med. The Cancer Center provides a lot of funding for student scholarship, funding for the actual research, and funding for graduate students and faculty members to travel to meetings. You are eligible for this funding as an undergrad as well, and I'd like to have some of the undergrads tell you about it. So I'm going to stop here and go to my lab and let you, while I'm doing that, I will let you talk to John Tumenberger, who is a undergrad with a Cancer Research Award. Thank you. Tell us a little bit about yourself, if you would, either in the chat or unmute and go ahead and talk for a second. So while you're thinking about that, I will introduce you to John Tumburger. I'm never going to say your name right. Great. Othniel. I'm glad to have you here. John is a junior, right?
Sophomore
Sophomore here at K-State, and he is going to show you a little bit of his lab and what he does in his lab. And then we'll show you a number of other people working in a lab and some things of what they do. So with that, I'm going to let John take over and share your screen.
Good morning. My name is John Tumburger, and I am an undergraduate researcher here at Kansas State University. First, let me start by introducing the lab that I work in. I work for Dr. Bradley Olson, and we study the evolution of multicellularity. Currently, I am focused on cell-cell adhesion and how things stick together. We use the volvocine algae as a model organism system. I particularly work with Chlamydomonas reinhardti, Gonium pectorali, and Volvox cautori. One thing that I have found to be especially great about research at K-State is mentorship. As an undergraduate, you have the ability to work with both PhD and master's students, so people who have already graduated from undergrad and are pursuing higher education. And through this, it has been awesome. I work with a PhD student currently, and my mentorship experience has been great. As a first semester freshman, I relied pretty heavily on the people in my research lab. It was really awesome to have people who had already been through the material that I was covering and were able to explain it in a different way. Another thing that I have is PI mentorship. So working with Dr. Olson has been awesome. We meet once a week. Not only is he my academic advisor, but also my advisor in the lab. And so we have pretty open discussions about my classes and also my lab work. And it's really awesome to be able to work with a faculty member so closely. Another awesome thing about working in a lab is the grant opportunities. So there are grants available through K-State and the Office of Undergraduate and Research and Creative Inquiry, as well as the Johnson Cancer Research Center to help fund your research. And so it's been awesome to be able to apply to those with the help of Dr. Olson and also the other members of my lab. So the impact on my undergraduate career that research has had is great. So I plan to have a future career in medicine. I plan to go to medical school. I'm a pre-med biology major right now. And I think that having a background in research is going to help me greatly as I go on to the next step. Another thing that I have really enjoyed is the application of classroom knowledge. So last semester I took genetics, and going through things in class gave me a good understanding of what was going on, but being able to apply those principles and complete protocols like PCR or plasmid extraction and things like that that we were talking about in class, and learn a little bit more about them on a hands-on level was really awesome. Another thing that undergraduate research can do for you is have your name included in publications. So you can directly contribute to furthering scientific knowledge, which I think is so cool. And the last thing was learning the scientific method. In every science class I've ever taken, the scientific method is always covered. But really applying it in the lab and designing my own experiments has helped me to better understand it on a deeper level. So that's a little bit about me and the lab that I work in. Now I want to talk about a procedure or some techniques that I use in the lab. So we all know that DNA is small and we can't see it with the naked eye normally. So how do we solve for that so we can see DNA? Well, using electrophoresis in an agarose gel, you can visualize DNA on that gel. So electrophoresis is a procedure that uses electricity to pull DNA fragments through the pores of an agarose gel. DNA is negatively charged, and so it will move through the gel towards the positive pole of your power supply. This is really cool because you are able to see the DNA fragments as they spread apart, and then using UV light, you can illuminate them, and you can visualize the DNA looking at the fragments by size. So the smallest fragments are going to move the furthest towards the positive pole, where the largest will stay towards the top. And using a defined ladder or a defined set of DNA, you can tell exactly the size of the DNA band that you're looking at. This is important because you can see sets of base pairs or sets of DNA in an agarose gel that you would not normally be able to see with the naked eye. Does anybody have any questions about research as an undergraduate or working in a lab or opportunities at K-State?
Okay, both Neil, if you have questions, go ahead and unmute and you can ask. What major are you in, John?
I am biology, and currently I'm thinking about adding a chemistry double major, but I'm also pre-med and a leadership minor. K-State offers great programs and it's easy to get involved wherever you want on campus. So biology is my first home and I really love it, but taking chemistry classes has given me a little bit more, I'm able to see a little bit more and do a little bit more. And so I also have really liked chemistry. And so I'm thinking about adding that double major here pretty shortly. As a pre-med, you get a lot of great opportunities. We have advisors as well in that department who are like great at helping students get into medical school. And then as a leadership minor, we talk a lot about how to be a leader in the real world. So I am interested in pursuing a career in medicine. Right now, my biggest focus is internal medicine, but I'm also interested in research. So whether that would be as a PhD or as an MD, I'm not sure yet, but I'm really interested in the patient care and human health aspect of research.
Okay, thank you, John. If you'll hang out for a little bit, we'll come back at the end with more questions. I am now turning the camera around and Molly is going to get some cells out of an incubator that we want to show you.
I just want to point out we have another guest in case you didn't notice.
Good morning, Caleb. If you have questions, either unmute and ask or type them in the chat. So Molly has 2 plates and these are melanoma cells. when they start out, can you tip that one so we can see the media? The media is quite red, but as they grow, the media turns much darker and almost to a black if you let it go long enough. She's going to move them to the microscope, and we will show you why they're different. At least we hope we will. I think that's it. Thank you. We have a light. We have a black screen. There we go. Let me see here. These are happy cells. They are growing quite well. And it's hard for you to see, I'm sure. Just a minute. But they're pretty shiny and clear. But if we look at the cells that have turned very dark, these are melanoma cells, and so the melanocytes are the cells that will grow, that allow your skin to have skin color, but they also can overgrow, and you can maybe see here at the top that we have some that are turning dark black. These came from a black-skinned mouse, and eventually these cells will turn very, very black. Of course, I can't find them right at the moment. They were there earlier. Depends on where in the plate you are. There's one, if I can get it to hold still.
This
Okay.
monitor is showing what's on your microscope right now?
This monitor is showing the microscope. Over here, I've got it under the microscope, and it's coming over to the monitor so we can try and show you. And you can see up here at the top, there's a dark cell and more dark cells and more dark cells. Those cells have differentiated into true melanocytes, and they will quit growing. So we really don't want to keep them at that dark stage. We want to keep them at the younger stage that you see in the rest of the screen. In order to do that, Devante is over here in our tissue culture hood, and he is going to be splitting cells. So Devante.
Hi everyone, I'm Devante Hammer, and like Dr. Fleming said, I'll be splitting the B16F10 melanoma cells right now. So I'm going to go ahead and take off the old media.
Just a minute, Devante, I want to interrupt. Okay. Are you a graduate or anything like that?
I'm an undergraduate studying microbiology on the pre-med track, and I'm a junior currently.
And he's working in the lab and doing a lot of experiments. You'll see some of his results at the end. Okay, Devante, tell us what you're doing.
All right, so I'm taking off all this old media. Now I will be washing the cells in TCPBS.
He's going to wash off the media because it contains inhibitors that allow the next step to not work. These are things that you will learn to do in the lab.
I'm going to take off TCPBS. Now I'm going to wash or kind of break up the cells a little more from the plate using trypsin diluent.
If you notice, he's working in a hood with a barrier so that everything inside that hood stays sterile.
And I'll be adding some new media too, and this will allow me to take the cells off the plate, and then I'll be able to centrifuge the cells down so we can collect them.
So the trypsin cleaved the cells off the surface, and now he's just stopping the trypsin with the media because it has trypsin inhibitors in it, and then he's going to put it into a tube. He's now going to centrifuge that tube for about 4 minutes. So during that time, do you have any questions? If you do, either unmute and ask or type it in the chat. He's balancing the tubes with a balance tube, and then he will centrifuge it. Okay, Devante, do you have anything else to say to them while it's going?
I got interested in research mainly because I wanted to apply some of the stuff I was learning in class, as well as kind of get to know small people and faculty and staff. And Dr. Fleming allowed me that opportunity, and I really encourage doing that. I personally had the K-Embry scholarship, and it provided funding and allowed me to be in the lab, also learning a lot of science.
It also has allowed me to get to know Devante quite well, so that I can write strong letters of reference for him when he goes to apply to medical school. I think at the moment, while we wait for 4 or 5 minutes, if you don't have questions, Devante, how long have you been in the lab?
This is my first year, and so just these past 2 semesters.
He's been in his entire junior year, whereas John started his sophomore year. It gives him an extra year. We can stop now for a second and show a video of Elizabeth. Stop share. I think, maybe. Okay. Here we go.
Hello, everyone.
Wait,
My name is Elizabeth.
wait. Okay, we're going to start that over. There we go.
Hello, everyone. My name is Elizabeth Ripper-Jetty, and I am a senior here at Kansas State University studying biology pre-medicine. I work as an undergraduate researcher in the wellness lab, and we study the effects of beta-HPV. I want to thank you for your support in allowing me to do fantastic projects like the one that I'm about to show you today. So today I'm looking at chromosomes by using this light microscope. If we zoom in high enough, we can see the DNA at a molecular level, and that allows us to see the damage that beta-HPV causes. Hopefully, this research will allow us to better understand beta-HPV and its effects on cancer.
Okay. Any other questions? How common is it for undergrads to join research labs? I think that we have not enough labs for all the undergrads, but we would like to have every undergrad have the opportunity to work in a lab, because it really helps their academics as well as letting them get some hands-on experience to understand what's really going on in their lab or in their classwork. So we get lots of them. Right at the moment, I only have, well, I kind of have 3. The Cancer Center gives usually about 40 to 50 scholarships. The Kinbury gives 20 to 25 scholarships. The Undergraduate Research Center, or OurC, gives somewhere between 20 and 50 scholarships every year. So there are a lot of them available. You can also work in a lab for pay. Molly over here is not currently on a scholarship, but I am paying her to work in the lab. She will talk as soon as Devante's things get done. You can also work in the lab for class credit. So you don't have to get a scholarship. You can do it for credit, and I highly recommend. Molly, how many credits have you taken for working in the lab?
It will be 5. I'm currently in 2, and I did 3 in the past, and they count towards my major as microbiology credit hours, so it's been really helpful in completing my major.
So she's currently working in the lab. She's taken a lot of credits by working in the lab, and she was a cancer research awardee a year ago. So there's lots of different possibilities. And I highly encourage you to check out the lab, see where your interest is, and and see if you can find a lab. Devante's done centrifuging.
All right, so now that I got that, I will go ahead and dump off the old media and resuspend the cells in new media and go ahead and plate them on the new plates, which I've already pre-poured media on.
So each of these pipettes, the long sticks that he's using, are sterile, and he takes them out sterilely and applies everything sterilely to the cells. Bacteria would love to grow in this media, and we're trying to prevent them from growing. But these cells that he's using are actually tumor cells. They're the same cells as what you saw on the under the microscope. And we can take those, and I will show you towards the end a picture of what happens if we inject just 1 million little tiny cells. You get probably 3 million off of one of these little plates. 1 million of those cells into a mouse, which forms a little tiny bubble, kind of like what you would see if you got a TB test and they made a little bubble. Go ahead, Devante.
And now that I got them all seated on, I will go ahead and put them in the incubator, and then I will do this again on Monday.
So he does this usually Monday, Wednesday, and Friday. We've made it special for today, but every week these cells need to be fed and taken care of. Okay. Other questions for Devante? Thank you.
Thank you.
So then we're going to come over here to what Molly's going to be doing. She is setting up an assay. So Molly, why don't you tell us a little bit about it?
This is a tumor protein assay. So I'm putting specific amounts into each well of this, and then we can put it in a machine that will read it and tell us information about how much protein and which proteins are in there, and that'll help us learn more about tumor cells. So
right now she's making dilutions down the plate so that'll be her standard curve. It's a dark blue color, and we can add other colors to it of reagents. The color really helps identify what things you've put in the well already. So Molly is a senior. She's a biology and anthropology major?
Yes, microbiology and anthropology.
Microbiology and anthropology major. And she will be graduating in May.
Yes.
So she's been in the lab for 3 years?
Almost.
Almost, yeah. Questions for Molly or Devante, or general questions? If there's no other questions right at the moment.
Hi everyone, my name is Lake Winter. I'm a junior working at Dr. Yang's laboratory here at K-State, and I thought I'd show you what a Biohazard Level 2 lab looks like. So here we have a heating plate, a thermocycler, a centrifuge, another thermocycler, and then our chemical cabinet, which we have ordered from A to Z. This is what a typical table looks like. I'm using Josh's as reference. You can see the pipettes there on the table. I'm going to pan over to Harry's and show you what a desk looks like when someone's in the middle of a pretty long experiment. Like most labs, we have an incubator, a heating plate, a weight scale, and an enclosed weight scale for more precise measuring, and a microcentrifuge, in addition to several other centrifuges we have in the lab. We also have a number of chemicals for mixing components together. We also have a shaking incubator, as well as a 4 degrees Celsius lab fridge that we usually store our infection media in. We also have gel electrophoresis kits, as well as a UVP viewer, which allows us to see the highlighted bands. Also to the right of us, we have the chemicals used for making Western transfer gels. Here's some footage from the next day of me working on a gel electrophoresis. You can't really see my samples all that well, but I have them in an ice bath. I also have the order of the ladder recorded on that sheet of paper there. It's about halfway through its run time, so I'll be coming back in about 20 minutes to take care of it. I'll put it into the UVP viewer and we can get a clear picture. We have a negative 80 degree fridge, as well as a liquid nitrogen storage unit, one of 2 we have in the lab. And we also have a negative 4 degree fridge, one of 3 in the lab. Moving on to the back room, we have a standard microscope, a sonicator, a luminescent microscope, a fume hood that we use specifically for cell cultures, a wash sink. We also have 4 incubators, 2 for cell cultures and then 2 for those infected with virus. We also have the hood we use specifically for cell lines infected with virus. And that pretty much sums it up. Thank you.
So the last thing I want to finish up with is some pictures here. This is a tumor that was taken out 10 days after we injected all those cells. And you can see all the little lumps and bumps. We injected it into a mouse, and 10 days later, it's about 1.5 centimeters long. We are interested in looking at what kind of white blood cells can be found in there. These are some of the white blood cells that we would like to stimulate. They're called macrophages. We want to stimulate them to phagocytose or eat those tumor cells. We also work in an intestinal model, and so here and someplace, and here we have some of the villi from our intestine, and we've stained them here on the right with a marker to identify the epithelial cells, so that layer that surrounds the villi, but also the white blood cells that are in the blood vessels. And then on the left, we've stained them for white blood cells and for the nuclei of the villi, so that we could tell each individual cell. You can see that some of these white blood cells are starting to crawl out of the blood vessels and into the epithelial layer. Okay. And all of these were performed, all of the staining and pictures were taken by undergrads. Undergrads also do the entire tumor model. So with that, I will take any additional questions that you have. We'll turn this around and see what kind of questions you have. Anything from you, Othniel? Magna? I don't know how to pronounce your name, so you're going to have to help me because I slaughter it.
Pronounced Othniel Maganga.
Othniel Maganga, thank you. Do you have any questions?
No, not at the moment.
Okay. What are you looking at doing for going to school down the road?
I was looking to go into pre-medical studies and go into med school. And
Okay.
I want to chase a career in being a neurologist.
Very good. As you heard, many of our scholarship awardees are pre-med, wanting to know more about the body. John, Molly, and Devante are all pre-med, plus they have their biology or microbiology major, and sometimes John's really ambitious with a double major, and they have a lot of outside activities and things that they're doing in their life as well. So it gets really busy, but it can be a lot of fun, right, John?
Yes, I would have to agree. So outside of the lab, I also volunteer. Currently, I'm volunteering for the Red Cross, but in the past, I've volunteered at local hospitals in Kansas City, as well as I'm the philanthropy chair for the Pre-Med Club and the mentorship chair for the Department of Biology.
So busy, but you have lots of time to prepare yourself, and we welcome you to get involved in research and come check out the Johnson Cancer Research Center's webpage and follow us on social media, and that can really give you an insight into what's going on. Okay. That is all that we have today. Marcia's putting the website in the chat, so you'll have it. If you have any questions that you think of down the road, send us an email or check out our website and see what you can find. Thank you for visiting.
Should I end the meeting or do you want to?
I don't think I can.
Okay.
I don't know how it was on the website, the other website.
Okay.
Do we?
It's gone now.
Yeah.
So I'll stop recording
Uh-huh.
first. There we go.
Thank you, John, for